right away. Our first speaker this morning is Dr. Don Smith, rooted in resilience, harnessing rhizosphere biology and stronger crops. Dr. Don Smith is a distinguished McGill professor, CEO of Biofuel Net Canada, and head of Biomass Canada. During his 39 years at McGill, he has supervised 86 graduate students, the majority of which were acquired PhD level study. Mr. Smith has published over 410 peer-reviewed papers and has 15 issued patents. He's worked largely in production and physiology of crop plants, more recently with an emphasis on plant-microbe interaction. So let's welcome Dr. Don Smith to PEI.
Sorry, I like to time myself, so otherwise I can get off on a tangent, etc., and waste a lot of time. So I don't want to do that. So, all right. Okay. All right. So let's see if we can make this work. So yeah, we're going to talk about plant microbes and their associations. So okay, let's see if I can do this correctly. Nope. All right. Yeah, okay, there we go. Thank you. All right, so yeah, so this is, we're talking here about the associated microbes. So plants, mammals, all of us, we have associated microbial populations. They say, for instance, that if you were to suddenly disappear, but your skin microflora remained intact, people will be able to recognize you. There's a lot of it there, so we all carry this stuff around. In the case of plants, we refer to it as the phytomicrobiome because it is with the plants. The phytomicrobiome plus the plant is the holobiont. And these guys have been together for about 500 million years since plants came on land, and arguably for a little bit before that, but that's more tenuous. The community of microbes is associated with all parts of the plant, but it seems it's more prolific, et cetera, more diverse around the roots because the roots are in the soil, there's moisture, there's organic matter, so there's a lot of scope for development of the community. They help in a wide range of ways with the plants. They help them with nutrient mobilization, hormone production or hormone analog production, disease control, and signals, which we work on a lot. So these work at very low concentration, so I sometimes refer to them as hormones of the holobiont. Am I doing something wrong? Okay, fair enough, okay. I'm not talking out of my forehead after all, so yeah. Okay. The line of research, so it started with soybean. Soybean evolved and was domesticated sort of south coastal China, so in the tropics and subtropics, and we agriculturalists have carried it both north and south ever since. Its metabolism is pretty much set up for high temperatures. In fact, the optimum temperature, for instance, for soybean nodulation has been known for over 100 years, and it's sort of in the 25 to 30 range, so pretty high. Soybean production in Quebec, where I am now at McGill, started in the late 1980s. I went there myself in about 1985, and so the soybeans came there about the same time I did. When we started growing them there, we noticed something unusual about the way they started out. They would come up out of the ground, they'd be nice and dark green. There's lots of protein and so lots of nitrogen in the seeds. But once they exhausted that stuff, they'd start to turn lighter and lighter green. They'd go almost yellow sometimes. And then suddenly they turn dark green. I was looking at this in the field, I was going, something is messing with the nodulation and the onset of nitrogen fixation. These guys are getting quite nitrogen impoverished. So we wondered if it might be low soil temperatures. I mean, the optimum temperature for nodulation is 25 to 30. Soil temperature at planting time in Quebec is usually about 10. So that's well below that. So yeah, we did some testing. We were able to show, am I doing this right? I'm just not that trainable. Here we go, okay. There we go, yeah, we're able to show that for each drop in temperature, soil temperature, the completion of nodulation, the onset of nitrogen fixation was delayed by a day or two. Once you get down below 17, so that's 20 to 17. Once you get down below 17, it was about a week or 10 days. And if you got down to 10, it just never happened. So there was something going on there with temperature. We did some testing, and we were able to show that eventually that the most sensitive part was related to the first 12 hours. And we were lucky because they were just publishing what was going on in the first 12 hours. What's happening at that point is signal exchange. So the plants are producing signals, isoflavonoids, to the bacteria. The bacteria senses the correct ones, then a bunch of nogenes come on, and they produce signals back to the plant. So they recognize each other as not hostile, potentially beneficial. So the plant allows the infection, et cetera. So we did it to try to overcome the problem. We just would produce our cultures. So we've got maybe a liter of liquid media that's grown the bacterium, and it's in the lab. We were going to use it in the field the next day to inoculate soybean plants. So before we went home, we would add the right amount of genistein, which is one of those isoflavonoids, to it, and just leave it overnight. So by the time you come in the morning, all the nogenes are on and things like this, and they're making the return signals. We just use that as an inoculant. And it had a very clear effect. So you can see here, where am I going here? Here we are. Now we're, okay. But anyway, you can see here, there's two different cultivars. This one is strongly affected. This one was still just starting. But by the end of the season, the amount of nitrogen fixed was increased by 80%. So pretty dramatic. And the yields were up about 20% usually on average. So it had a very profound effect. I looked at this, and at the time we were going through a recession, and I had a really good lab manager, and I was worried I would be in a position where I couldn't pay him. So what I did was I looked at patenting the technology. It was okay, I could do it. So we patented it, and we set up a company called Bios Agriculture, and then he was the president, so he had a job, right? It was good. So he was the head of Bios. Bios was eventually taken up by a larger Canadian company, Agribotics. Agribotics pushed the technology closer to the marketplace and then sold themselves to EMD. If I remember correctly, they sold themselves to EMD for 32 million in euros. That was 32 million euros. And so EMD sold a chunk of themselves eventually to Novozymes, and that chunk included our technology. Novozymes hardwired themselves to Monsanto. Monsanto was swallowed by Bayer. And so it's a corporate food chain, right? It's an interesting situation. But Bayer, I was talking to a guy, I was with a guy from another smaller company. We were talking to a guy who just retired from Bayer about 6 years ago now. And he had been in the division of Bayer that actually had our technology, and he indicated that at that time Bayer had made about $1 billion on it. And so now maybe $2 billion, I don't know. But anyway, they don't tell me these things. So, all right. So this is sort of another thing that came out of all this. When we were testing this stuff in the field, we would put the genistein in, the isoflavonoid, and then we get production of these signals. But when we did that, we were noticing that the plots that had those treatments the seedlings came out of the ground faster, which was surprising because the whole nodulation thing was after that. So I actually was having a long discussion in an empty classroom with one of my PhD students, and we set up an experiment. Here we are. The simple thing is germination, seeds in water, genistein by itself, so that's the signal from the plant, Bradyrhizobum japonicum alone, or Bradyrhizobum japonicum with genistein, which would cause the Bradyrhizobum japonicum to make the LCO signal. Am I too embedded in technological terms and acronyms here? You guys should tell me if I am, because anyway, all right. So, oops. Yeah, so we did that. And what we found was that, and actually it was one of those things with the student, we had it all organized. The student was just going out the door, and we were talking about soybean, but corn also has a problem with low temperature in the spring. So I said, you know what, try it on corn. It almost surely won't work, but I like my PhD students, I like them each to do at least one mad science experiment before they finish. So this was his. But interestingly, it worked on corn. so this was a total surprise. You can see here the difference in the size of the corn plants. And it worked on almost every plant we tried it on. So I would argue probably the role of this LCO in, I keep doing that, of the LCO in signaling to the plant and causing growth stimulation was probably older than the role in signaling in the nodulation. Nodulation is only about 1 million years old. Sorry, about 10 million years old. But anyway, so this is what it looked like. You said you could see a number of these other plants were affected. And so we were doing a lot of work on this, and we were commercializing it a little bit more ourselves. So we were concerned about the jasminates. These are the signals from the plants. These guys are expensive. At that point in time, they were up to $1,000 a milligram. And at the same time, they were at higher concentrations, which is what we needed for this. They were not particularly nice to the bacteria. I mean, these things are produced as phytoalexins, so they're produced to make other organisms that the plant doesn't want around have a difficult time. So they're tough for living things. My students came across a paper that suggested that jasmonic acid could do the same thing. So here's jasmonic acid. And so we tested it, and it seemed to work well. The pattern of, here we are, this is one of the key genes in manufacturers of the LCOs. And this is addition of genistein, and it's as you'd predict. It comes up quick, and it comes up to a good level. But when you add the jasmonate, it comes up more slowly. So you need higher concentrations, but it's so cheap it doesn't matter. And it went up higher. So this was kind of interesting. We filed a patent on this, and we moved on commercializing it. It wound up initially with an American company called Becker Underwood, and eventually with BASF. I'll explain how that happened in a minute. This is what it looked like in the field. So we've got, this is no inoculation, no bradyrhizoma japonicum nitrogen fixing bacteria added. This is a soybean with them added by themselves, them added with the genistein, and this is them added with the jasmonic acid. And this is the two together, so no obvious synergies here. And this guy is doing better than this guy, there's no question. So this was interesting and potentially a better mechanism. So what McGill had done is they had licensed one technology to produce LCOs to Becker Underwood, and another one, effectively at this point, it was with EMD, or maybe it moved on the next step, I forget. But in any case, what happened then was, it was Novozymes by this time. And what happened was that Novozymes, Becker Underwood was a smaller company. They were marketing hard, and they were starting to take market share away from Novozymes, and they didn't like it. So they sued Becker Underwood. They sued for patent infringement. So all the patents were out of my lab. So I got to spend a year in court, which was, I don't know, people will tell me things like, oh, you must have learned a lot. And say, yeah, I learned a lot, but I could cheerfully have spent my life not knowing that stuff. You know, it's just, you get into litigation, it's often the dark side of humanity. So anyway, so I spent most of the year in court doing depositions and things like this. And but in December, it was out of Wisconsin jurisdiction. The Wisconsin judge dismissed both suits, and BASF bought Becker Underwood. So that's the link to BASF. So they still have it. McGill audited Novozymes, and I think there was some cash came out of that. You're okay so far? Okay. So other PGPR, other signals. We went to the field. We looked at isolating other bacteria to see if we could find something else that was equally interesting. We isolated 14 strains from the roots of soybean plants, the nodules of soybean plants that were, you walk through the field, they're growing, you see individual plants that look like they're growing better than the ones around them, and there's no obvious reason for it, so we take those guys. And we had a set, there was I need this thing to work here. Oop. No, it's not gonna work. Anyway, oh, there it was. Uh-huh. Okay, is this okay? Aim it at this one? No, I'm not seeing it. Anyway, I'll carry on in my haphazard fashion here, it's all right. But anyway, so so there were three that were particularly effective, and one that was the most effective of all of them. And so we tested stuff from that it might be producing, and we call it fluorescent 17. This was partially isolated at this point. You could see clear effects on plant growth, corn, soybean here. And for me, I just assumed this would probably be something similar chemically to the LCOs, and that so I had a really, really good master's student came along at the time, and I said, it's probably LCO. So try testing for that first. So she did nothing, right? So she started doing general staining, and it lit up for protein. So this is samples coming off. These are active samples coming out of our HPLC. And these are stained for protein. That's why you can see this. So it was clearly had at least some protein in it. But it wasn't LCO. It wasn't what we thought it would be. And so it turned out it's a peptide. It's a small protein. And it's also a bacteriocin. It was funny, the student, I said, this was a really, the student was very engaged. She called me up at about 11 o'clock at night one night. She was like out of breath. She goes, I think I know what it is. I'm going, really? What is it? She goes, it's a bacteriocin. I said, wow, that's exciting, but you have to tell me, what is a bacteriocin? These compounds are used as food preservatives, so it's like not something I knew too much about. Anyway, but you can see here, we tested it on 53 strains of bacteria, and it inhibited 32, generally the ones that are more closely related to it, and that's what bacteriocins do. They often make life difficult for strains that are closely related to them. And that makes sense, right? If they can remove those strains from the environment, if they're closely related to them, they're probably consuming similar things to the strain that's producing the bacteriocin, so they get rid of the most intense competitors. But you can see here there's inhibition zones around these ones. I'll never get this thing going. But anyway, yeah, inhibition zones, and not with this one down in the corner here. Ooh, there we go, not with this one. So this one it didn't affect. But so this is just a piece of filter paper with our Thyrsin 17 on it, and this is the background, the strain that is being tested against in the background. They're different colors because of the growth media that they grow on. Okay. In addition, what was interesting, this little protein was stable over a wide range of pH and temperature. For instance, you can boil it and it doesn't care. Right? If you autoclave it, you can denature it, but boiling doesn't do it, which is really not typical of a protein, right? So we've done some work on it since. It seems it has 4 thioether bridges inside it. So these stabilize it. Anyway, okay, I'll press on. So we looked at the genes that code for it. There are 3 genes. They're on a plasmid. They're 3 copies of the same gene. They've been separated long enough, they have evolved, right? But you start to realize this particular single compound does 2 things for the bacteria. Oh, look, they just turned off my stopwatch. Oh, look there. It does 2 things for the bacteria when you think about it, right? It helps the plant grow better, so it increases the niche space for the bacterium. It also removes the biggest competitors from the niche space. So it makes the niche space bigger and removes competition. So there's a lot of, it's constrained evolutionarily. Evolution doesn't want it to change very much. That's the wrong way to say that, but this is what happens. And so when you look at the 3 copies of the gene, they're not identical. They've been separated long enough, their nucleotide sequence, they've been separated long enough that they have evolved, right? But when you look at the proteins they all produce exactly the same protein. So every time there's a change, it's at the 3rd position in the codon, and it's one of those redundancies, they call them, where the change in the exact nucleotide there is such that it still codes for the same protein, the same amino acid. That's all that's been tolerated through the course of evolution. It's an interesting situation. All right. So one of the other things with all these, with both the Thyrsin 17 and with the LCO, was that when we were doing field work, it was fairly consistent. When we were doing lab work, about 1/3 of the time, it wouldn't work. So this is just, I spent 2 years pulling my hair out, going, what the heck are we missing here? You know, it's like, and eventually I was standing in front of a field test at our facility. It was a cold spring, the site was heavy clay, and we had treated the stuff, we had treated corn with the stuff, and the corn plants that had received the treatment were about, maybe a little over 10 centimeters out of the ground. The controls weren't out of the ground. So there was a very dramatic difference, and I was looking at this going, maybe it's about stress. I had just gotten a really good student into the lab at the time, so I got her to test this. And you can see here, this is at 22, which is for Arabidopsis. This is on Arabidopsis. You can see it's growing, they're all about the same. But here we are, Arabidopsis at 10, which is stressful for it. This is the control, and these are the ones that got the signal molecules, as we call them, treatments. So clearly better. Okay. This is just looking at some other stresses. So this is salinity stress. You can see this is no salt. Again, Arabidopsis plants. This is 150 millimolar salt, and you can see the plant's only about 1/10 the size. And here it is with 2 levels of LCO added, and pretty much restored the growth, and 2 levels of Thyrsin 17 added. This one, it's remarkable, this stuff, the concentrations. This is 10 to the minus 11th molar. It's not a lot, right? And when you look at it, it's like, well, I started doing checking for human hormones, right? The one that works at the lowest concentration is testosterone in females, and it's at about 10 to the minus 10. So it's definitely in the hormonal range. So that's why I started calling them hormones of the holobiont. Anyway, and this is signals for under water stress. So this is osmotically imposed water stress. Same thing. We did a bit of looking here at genes that were affected. You can see the largest increase was in stress response genes. These plants were already stressed. This is soybeans at 15. But when plants are stressed, they can often go one of two directions. They can try to grow through it or shut down. And this was maybe switching from the shut down to the try to grow through it genes. This is just looking at genes tied to energy. You know, we knew that stuff tied to stress were upgraded, but this is energy. We see those two, which explains why they grow faster. This is just looking at, I also thought that probably we're using the same receptor, but I think that's not true anymore too, because you look at these two hormones, this is plant hormones, going the same way, these two going the same way but at very different levels, and these two going in exactly opposite directions. So you guys, it's working by a different effect. So this is just them, hormones of the holobiont. They do lots of nice things for the plants. They're inexpensive, they're in the environment already, et cetera, but they're very poorly understood, and they seem to have a huge amount of potential. And I said the one we've commercialized already has done some nice things. This is just calculation of potential greenhouse gas reductions. So in Canada, it could be somewhere around 3 million tons per hectare per year. That's my last slide, so I think I'm at 20 minutes. Okay. Should I just leave? Well, I've used up all my time, or can we take a question? Anybody got a question? It's early in the morning.
Did you commercialize one
Yep.
of your compounds so far?
The LCO, yep.
Is it publicly available?
Yeah, it's in a lot of products. If you type it in on the internet, it's in quite a few products now. Yeah.
Can I see what it's called?
You know, honestly, I forget. But if you type, it's terrible. No, but Bayer, for instance, produces about 20 different products with this stuff in it. Yeah, so if you may well be buying stuff with the product in already. It's widely. I say they made a couple $2 billion off it, and I think one of my grad students, I don't know how accurate it was estimated it had gone on in each of the last few years. It had gone on something like 100 million hectares of agricultural land globally. So I don't know. Yeah. Anyway, anybody else? Okay, can I take one more? It's up to you. Okay, go. So.
Are you still continuing to discover new compounds like this today? And
Yeah, we are. We're doing a large-scale screening right now, and we start by we'll do the initial work with the strains, then we'll grow the strains in liquid culture and remove the cells completely, and then see if the liquid cultures require the ability to stimulate seed germination. It's just a quick test, and if it does that, then we assume there's something in there. Then it's a long. You can isolate peaks, and in one of the cases we looked at identification of compounds. I'm willing to do it, but our company actually has to pay. It took us 3 years to identify one. So it depends on what it is, but it can be challenging. So anyway, I should go. Yeah.